Celprogen bases its excellent reputation for quality and product purity by adhering to a strict regimen in the lab. This is the procedure for cryogenic preservation of cells by Celprogen. Prior to freezing, the culture should be maintained in an actively growing state to ensure optimum health and good recovery. Ideally, the culture medium should be replaced 24 hours prior to harvesting. Using a sterile pipette, remove and discard the culture medium. Any materials and solutions coming into contact with cells should always be disposed of properly. Rinse the cell monolayer with 5 to 10 milliliters of sterile 1X PBS solution to remove all traces of fetal bovine serum. Add 3 milliliters of trypsum EDTA solution for a T25 flask and 5 milliliters for a T75 flask. Incubate at 37 degrees Celsius 5% CO2 in a humidified incubator for 2 to 3 minutes. Check the progress of the enzyme treatment every few minutes on an inverted phase contrast microscope. Add 12 milliliters of serum-containing growth medium to deactivate the trypsum solution for a T25 flask of cells and 10 milliliters for a T75 flask of cells. Vigorous pipetting may be required to wash off any remaining cells from the bottom of the culture vessel or to break up cell clumps into a single cell suspension. Collect the suspended cells in a 15 milliliter centrifuge tube. Centrifuge the remaining cell suspension at approximately 100 G for 5 minutes to obtain a soft cell pellet. Count the cells from the sample you took prior to centrifuging the cells using a hemocytometer while the suspension cells are being centrifuged. Use trypan blue solution to check cell viability. Remove the supernatant media from the centrifuge cells and resuspend the cell pellet in enough of the cell freezing medium to give a final cell concentration of 2 to 5 million viable cells per milliliter. Label the appropriate number of plastic cryogenic vials with at least the name of the cell and the date. Finally, add 1 to 1.8 milliliters of the cell suspension to each of the vials and seal. A slow and reproducible cooling rate is very important to ensure good recovery of cultures. When freezing cells, a decrease of 1 to 3 degrees centigrade per minute is recommended for Celprogen cell culture. Securely store cell cultures in the vapor phase of liquid nitrogen. This concludes the Celprogen cryogenic protocol for the preservation of cell cultures. If you have any questions or suggestions, please contact Celprogen customer service at area code 310-542-8822 or email customerservice@celprogen.com.